So send me an email, I'll fix it for you.
Yes, ma'am.
I will fix it for you. Yep. Okay. Where's my blank? Yes, ma'am. Getting them back on Friday. Getting them back on Friday. I know, but it will be Friday. Okay, so you can go ahead and do your seating chart. Let me put in iClicker. I know I haven't done that yet. I'm like a minute or two behind today. Just one or two. Just one or two. Okay. I think, and we ended on, we were talking about disaccharides, right? Okay. Okay, now iClicker is up. Seating chart is up. Okay, now we're rocking and rolling. All right, so today we'll talk about how we make glycosidic bonds, right? Because we talked about all the monomers before, but now we're going to talk about disaccharides, and then we'll talk about how we use glucose in metabolism, which is awesome. Okay, disaccharides. All right, I'm trying to remember the joke from today. Yeah, no. But did I tell y'all where does spaghetti go to dance? To the meatball. To the meatball.
I don't know why, but my brain went to it all. I was like, no.
Okay, so we've talked about our sugars, right? And we talked about if we have the alpha anomer and the beta anomer. How do we know the difference between alpha anomer and a beta anomer? Alpha is one up, one down, right? What's up, what's down? The highest number of carbons. The highest number of carbons is always going to be up. And then we're looking at the hydroxyl. Is it up or is it down? If it's down, it is alpha. If it is up, it's beta. Okay. So that initial position of the hydroxide, whether it's up or down, is going to determine how these monosaccharides can create disaccharides. And so you're always going to link them through an O glycosidic bond. What does that mean, an O glycosidic bond? There'll be a sugar, an oxygen, a sugar. That's a disaccharide, okay? So it's always a condensation reaction. So whenever we talk about a condensation reaction, what do you make in a condensation reaction? You make water. Why do you make water? Because you're starting with something that has a hydroxyl, right? You're adding something else that has a hydroxyl. Doesn't matter what it is. This is general condensation reactions. What happens? So you would take, it doesn't matter which one you take it from, an oxygen and a hydrogen. So you take a hydroxyl and you take a hydrogen. Hello. And a hydrogen. And from that, you take this away and you make water. And then what's left? R, O, R. That's an O glycosidic bond. What are the R's? The sugars, right? The sugars. So there are three really common disaccharides that you need to know. Lactose is one of them. Lactose is galactose, right? It's a disaccharide, so we have to have two monosaccharides. And it's in a beta 1,4 glycosidic bond with glucose. So we're going to look at what these are, but you need to know some of these. So then sucrose. Sucrose is what we have is table sugar. Table sugar. So if you're going to bake a cake, you're going to use sucrose. So when you use sucrose, it's going to be glucose, and it's going to be an alpha 1 beta 2. So it's going to be glucose, 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 and it's going to be fructose. And then our last one is found only in insects, Trellahose, and this is glucose that is in an alpha 1 glycosidic bond with another molecule of glucose. So do you see in those names, some of them only have one designation of alpha beta and some have two? Do you see that? Where we have beta 1, 4. There's only designating one in the alpha or beta configuration. But look at the next one, sucrose is glucose 1, alpha 1, beta 2 to fructose. Do you see that? Okay, why is that? So let's look at our common disaccharides. So when we look at our common disaccharides, here's lactose. The way that we name a glycosidic bond is you, number one, find your anomeric carbon. Where on this first sugar is my anomeric carbon? Right here, right? Okay, this is my anomeric. How do I know it's anomeric? It's participating in how many bonds to oxygen? 2. Okay, that's how I know it's anomeric. Now, I have to look, is there a carbon that's attached to the anomeric carbon that's outside of the ring? No, right? So this would be an aldehyde. This came from an aldehyde, not from a ketone. Yes? If it came from a ketone, that carbon that would come down, it would be like CH2OH. This would be carbon number 1. Right? You have to give that carbon number 1. But in this case, because there is no carbon outside of the ring attached to my anomeric carbon gets to be number 1. Now, which carbon is participating in the glycosidic bond? Is the anomeric carbon participating? Yes. If the anomeric carbon participates in the glycosidic bond, you name it as alpha or beta. So I'm going to look and I'm going to say my carbon number 6, which is outside of the ring, and my glycosidic bond. Are they up and down or up and up? They're up and up, so this should be beta. Now, my anomeric carbon is carbon number 1, and it's participating, right? So that's why I put the beta. Now, my next sugar, which carbon is participating in the bond? So I find my anomeric carbon. If there's a carbon attached to it, I give it a 1, but there's not. So I go 1, 2, 3, 4. So carbon 4 is participating in the second sugar unit of the disaccharide. Is the anomeric carbon participating in the bond? This one right here. No. So do you give it alpha or beta? No. So this is a galactose to glucose beta 1,4 glycosidic bond. Yeah?
So since it's not the anomeric carbon or the last one,
Correct. So let's do all of these. Like the answers are here, but unless you try it, right? Okay, so look at your first sugar. Find your anomeric carbon. Where's your anomeric carbon? Right here. What carbon number is that? Number 1. And lo and behold, it's the one that's participating in the glycosidic bond. So now we have to name it. So is it alpha or is it beta? It's alpha because this is up and this glycosidic bond is going down. Okay, so we have an alpha. Here's the 1. Now go to your next sugar. Where's your anomeric carbon? Be careful with this one. Participating in 2 bonds with an oxygen. It's right here. Now you've got to be very, very careful. This is not carbon 6. What carbon is this? This is carbon 1 because it's bonded to the anomeric carbon. So this is carbon number 1. This is carbon number 2. 3, 4, 5. This is carbon number 6. Remember how I said the standard is to write carbon 6 being up, but will you see it sometimes differently? Yes, you will. So you have to be careful. Carbon 6 is going down. How is my glycosidic bond going? This is going down. What's my glycosidic bond doing? Up or down? It's going down. So down and down is beta. Now name the carbon that's participating in the glycosidic. 2. And then you put a little arrow between them and put it in parentheses. So the reason I have alpha and beta here is because they are both of the sugars have the anomeric carbon that's participating in the glycosidic bond. Yes? Okay, let's do the last one. All right, look at your first sugar molecule. What do I have? Anomeric carbon, give it number what? Give it number 1. It's participating in the glycosidic bond. So do I have to give it an alpha or beta? I give it, I do. So I look at my carbon number 6 and I say it's going up. My glycosidic bond is going down. So it must be alpha, and then it's carbon number 1. Okay, now what is it participating with? Well, I got to go find this. Oh, this is a little weird, right? But find your anomeric carbon. It's bonded to 2 oxygens. That is carbon number 1. All right, now, where's carbon number 6? You can't go to the oxygen. When you're numbering carbons, don't go to the oxygen next. Go to the carbon. So 2, 3, 4, 5, 6. 6 is outside of the ring and going what? Number 6 is going up. So if 6 is going up and my bond is going down, I am alpha. And what carbon number is participating? 1. So this is glucose and glucose, which there are specific abbreviations for glucose. Glucose is GLC. But it's a glucose to glucose, alpha 1 glycosidic bond. Think you could do that on a test? Awesome. Yes. Can I what? Sure. Sure. Absolutely. Absolutely. All right. So now, a lot of times you eat things that are not straight glucose, right? You eat things that are disaccharides or polysaccharides, right? And so when you eat them, your body has to hydrolyze them. So you have to have the enzymes present in your body in order to hydrolyze particular disaccharides, right? So remember, hydrolysis is going to be the opposite of condensation. In condensation, we made water. In hydrolysis, we're going to hydrolyze, we're going to break water, right? So basically what happens is you have 2 individual sugars, right? They're linked by the O-glycosidic bond. So what happens is one of those glucose molecules is going to, well, one of the sugar molecules, it's not always glucose, is going to keep the oxygen, right? And then it's going to get instead, right? If you took this whole thing, you would get the oxygen and you would pick up a hydrogen from water. And then this one you would get rid of this, would pick up a hydroxide. So then you would end up with two monomers that each, instead of having a glycosidic bond, now have hydroxides, right? And that's exactly what we got over here. So you have to be able to go both ways. Can you make the disaccharide and can you make the monosaccharide from the disaccharide? Same thing here with sucrose. All we're doing here is now you're making two different sugar molecules. This one was glucose and glucose. Now we're making glucose and we're making fructose, right? And then you have lactose, it's the same thing. But these are specific enzymes. And there are enzymes that we don't express equally throughout our entire lives. What is one of those enzymes that we don't express all the time? Lactose. So most people, as they get older, become lactose intolerant. Why do you become lactose intolerant? You don't make the enzyme that can break down lactose, which is lactase. You don't make it anymore because you don't need it anymore, right? And so that's why people become lactose intolerant, right? So it's expressed, lactase is expressed in high levels as an infant always, so that infants can digest lactose. But as we get older, it declines. And so then you end up, you know, there are multiple ways that you can get around this, right? You can take a supplement that has the enzyme in it. So like if you know you're going to have that bowl of ice cream, you take the Lactaid, right? And you can have your bowl of ice cream because it has the enzyme in it. Or you can buy milk that already has the enzyme in it, right? So it already does the hydrolysis, but people say it tastes different, you know? Or you can buy milk that is just doesn't have any, right? Okay. Yay, we're ready for glycolysis. Everybody good on sugars? Very happy? We all understand sugars? All right, so let's talk about glycolysis. But before we talk about glycolysis, let's talk about what's going to eventually happen. Like I want to go all the way to the finish line. So you eat some sugars, right? So we have glucose. So that glucose goes through glycolysis. Then what happens to that sugar? We're going to break it down into two pieces, basically. What is the next cycle? The citrate cycle. Krebs, right? Depends on what you learn terminology for. But I prefer citrate, and I'll tell you why when we get there. And we want to go all the way from glucose, right? CO, from glucose all the way to our end products, which are CO2 and water. So we have to go through glycolysis, then we have to go through citrate, then where are we going? Electron transport chain, but we call that whole thing oxidative phosphorylation. Y'all called it the ETC, right? Electron transport chain. So that's where we want to go between now and the end of the semester. That's our plan for the end of the semester. So what is glucose? What's the molecular formula? C6H12O6, right? And we have to do this entire process. We have to have what other substance present? Oxygen, right? Plants don't need oxygen. Plants produce oxygen. We, on the other hand, need oxygen, and we produce CO2 and I just gave it away, right? Water, right? But really this would be, we got to balance that, right? So in glycolysis, glycolysis itself does not require oxygen. It's only the processes that occur in the mitochondria that require oxygen. But overall, to make this complete oxidation of glucose to carbon dioxide and water, you will need to have oxygen present. So the actual delta G value of this is negative 2,938 kilojoules per mole. So can the body do it? Yes, we have a negative delta G value. Perfect. So let's talk in detail about this very first step of glycolysis. So glycolysis is what we call one of the core enzymatic, let me move this, the core enzymatic metabolic pathways. And the reason that we call it one of the core pathways is because this is really the primary pathway for ATP generation if you don't have oxygen present. So this is no oxygen. Where's a place where we don't have oxygen present? There's a cell in the body that requires tons of ATP, but does not have oxygen. Your muscles can work under no oxygen conditions, but not always. Red blood cells. Red blood cells have no mitochondria, so they don't have any oxygen. They have sacrificed their mitochondria, they sacrifice their nuclei, they sacrifice everything to basically be bags of hemoglobin, right? And we talked about how important that was. But the cell still needs ATP. And so you're going to primarily have it there, right? And then glucose, when it's metabolized in glycolysis, can make many different, its product can go in many different pathways. So the metabolite that is made from glycolysis, so the product that is made from glycolysis, can be used in all kinds of other pathways. And one of those pathways is mitochondrial ATP synthesis, which is where we're going for this class, right? And the other reason, so this is a core pathway because, and this is kind of the reasons, right? Is that these enzymes, these glycolytic enzymes, are so highly conserved. So when we say highly conserved, what does that tell us? That we, as very complex organisms, have these enzymes, and we have really high homology. So our DNA that codes for these enzymes is extremely similar between us and freaking bacteria, right? Something really, really, really ancient. So because of that high degree of homology, it means that that is a very conserved pathway. It's something we see in all kinds of different organisms, right? That's where sequence homology tells us how old a pathway is. And glycolysis is extremely old, extremely old. So when we talk specifically about glycolysis, we're going to start out with one molecule of glucose. And we're going to start out that one molecule of glucose has 6 carbon atoms inside of it. And when we undergo glycolysis, we're going to convert that glucose from a 6 carbon molecule to 2 3-carbon molecules. And those 2 3-carbon molecules are called pyruvate. So we get 1 glucose going to 2 pyruvate. There are 10 enzymatic reactions. Do you need to know the 10 enzymatic reactions? See my head nodding? Yes. Need to know them. Yes. So all of the reactions can be divided into 2 subgroups, stage 1 and stage 2. Stage 1 is called ATP investment. You got to think about it, right? Our whole goal of glycolysis is to make energy. And then we have these byproducts that we can use to make some more energy, right? But glycolysis needs to do something for us, and that is to produce energy. But in glycolysis, you have to put a little bit of energy out to get a little bit more produced, right? So we have an investment, and then we have a payout. The payout is in stage 2, where we're going to actually make ATP. So what you have sitting in front of you on your little sheet is this picture right here. I gave you this? No, it does. It's a PDF. So all the animations are linked on the Moodle site. So if you go to Moodle and you go to chapter 9, it'll be there.
No, it links to the Pyruvate one.
Really?
It doesn't. That happened with the last chapter too. I kept trying to click on the animations and they kept leading back to like the first animation. Chapter. Yeah, but it's not there. Like I looked at it and it's not there. Like when I click on it right now.
You should be able to log into your ebook, and there should be a tab that says like animations too. So there should be another way to get it. I'll try to look at fixing the links.
The phosphorylate and the pyruvate kinase.
So there are two tabs. One of them is molecular animations, and the other one is what's it called? Process animations. Yep. Yep. If you don't see it in your list, check the other tab. Yep. So I will look at fixing animation links. Y'all said it was bad in chapter 8 and chapter 9.
In chapter 8, I didn't think to check the tabs, so it might be in the tabs, but in chapter 8, I was just like it was leading back to just one of them, and I was like,
yeah, like a lot of them like to bring you back to one singular animation, you have to manually click on.
Okay. I will see if I can get that fixed. But because that's something that I can't link straight out because it has to go through the publisher because you have the subscription. So I'll see what I can do with it. All right. But you should watch the animation because the animation's really nice. Like I always say, the animation's really nice. Go watch the animation. Okay. So this is kind of an overview of what we're going to do for the rest of the semester, right? So we're starting here at glucose. And we're going from glucose to pyruvate. This is glycolysis. We're going to do this stepwise, right? So in glycolysis, we're in an anaerobic environment. What anaerobic environment are we in? We're in the cytosol. Cytosol does not have oxygen. Mitochondria has oxygen. So we're going to convert our glucose to 2-pyruvate. And when we do that, we're going to make 2 ATP molecules. So not very much, but we're going to make some. That pyruvate can have one of 3 different fates. So the 3 fates of pyruvate are being converted to lactate, being converted to ethanol and CO2, or being transported into the mitochondria and converted to acetyl-CoA. So it's going to depend on what organism you are in and then what subcellular location you are in, what's going to happen to that pyruvate, okay? So if you eat glucose, this is what happens. You're going to take your acetyl-CoA, you're going to go through the citrate cycle, you're going to go through oxidative phosphorylation in the electron transport chain, and you are going to make an additional 28 ATP in oxidative phosphorylation and 2 ATP in the citrate cycle. So how many ATP do we have now? Plus our 2 up here. So we have about 32 ATP. Now, I put the about because it depends on what tissue type you're in, and we'll talk about that later. The numbers are a little different depending on which one you are in. But you can generate lots of ATP this way. But this is not our only energy source, right? Most organisms will store energy in another way in the body. So we can store glycogen, right? That's the sugar storage. But that's not the only form of storage that we have. How else do we store energy? Fats, fatty acids. So you can have fatty acids, and fatty acids can be oxidized to produce the same intermediate of acetyl-CoA. So when we undergo fatty acid oxidation, you're still going to go through the citrate cycle and you're still going to go through oxidative phosphorylation, and you're going to make tons of ATP. So fatty acids are a high energy storage molecule. But typically, we don't do that unless we are not getting enough glucose, right? So that's the whole point of dieting, right? So then you force your body to go and use those fatty acids as that are being used as storage, right? Okay, so this is the handout I gave you. These are all the stages of glycolysis. So the very first part here, the ATP investment. The ATP investment occurs while we still have one molecule. When we go into ATP earnings, then we split glucose into two. So that's why you see it, the pathway diverge, right? We start off as one sugar, we phosphorylate it, we rearrange it a little bit, and then when we get to the end of the investment stage, we cut it. And then we have 2 3-carbon molecules that we do different things to in the end up with pyruvate. So we're going to talk about each of the stages, what's important and all that kind of stuff. But what you need to know is where are we putting in ATP? Where are we getting out ATP? So there are 2 investment points in stage 1. The first one is hexakinase, when we go from glucose to glucose 6-phosphate. So if it's a hexakinase, what does it do? It phosphorylates. So that's why we go from glucose to glucose 6-phosphate. Which carbon atom do we add the phosphate to? Carbon 6. Hey, they named some stuff well. All right, then we have a phosphoglucoisomerase. Are we going to add phosphates with an isomerase? No, what are we just doing? Just kind of rearranging, Now we have phosphofructokinase 1. We have another kinase. That kinase is going to do what? It's going to grab a phosphate from ATP and slap it on to our sugar, right? Now we have fructose 1,6 bisphosphate. So we now have a phosphate group on carbon 1 and on carbon 6, right? So these are our 2 ATP investments. Now, when you look down at the ATP earnings, stage 2, what we're going to see is we're going to see ATP in 2 places, right? Kind of early in investment and early in earnings and then late. So if we invest 2, so I guess that really should be a minus, right? We're using up 2 ATP here. How much do we produce down here? 4 for a net of what? Net of 2 ATP. That's why they put these ATP as like black are investment, red is earnings, but full red circle is basically the net that you're going to get, right? Okay, so let's look at these individually. So when we look at these reactions, remember we need reactions to have a negative delta G value, right? So when you're looking at this, can you see that there are 3 different reactions that are highlighted? Why are those 3 reactions highlighted? Well, they do have negative G values, but there are other reactions that have negative G values. So why are these highlighted in particular? They have the most negative delta G values, right? So the more negative a reaction becomes, so 1, 3, and 10, the more negative a reaction becomes, it basically becomes irreversible. Irreversible because we have a very negative delta G value. Do you see how all the other ones are really close to 0? Either they're a little bit negative or a little bit positive. That basically means that all of those reactions are reversible. So all the ones with delta G, if delta G is close to 0, that means that it is basically at equilibrium, right? It's reversible. Or at equilibrium. right? It's only going to go one way if you have a massive negative delta G. But if you have a delta G close to 0, oh gosh, it could go either way, right? So these irreversible reactions are highly regulated reactions within glycolysis because they are going to determine whether or not glycolysis is going to happen, right? So you'll see lots of regulation here. These are like the determining reactions. So these are really important reactions. So if we add up all of these values, then you can look at the overall delta G for glycolysis. Now, sometimes it's really hard to actually determine the real delta G value inside of a cell. It's not so bad in the cytosol, but once you get into the mitochondria, when we get to citrate cycle and all that kind of stuff, it's going to be even more difficult to get actual delta G values versus, you know, standard conditions, because it's hard to assay in those really small subcellular locations. So at equilibrium. All right, so in our very first reaction has technically two enzymes, either hexakinase or glucokinase. So glucokinase occurs in only specific organs of the body, so only in the liver and pancreas. EAS, pancreas? I don't know. What does the liver do in relation to sugars? Why is the liver really important? It makes what? Glycogen. Right? Why is the pancreas really important in regulating blood sugar? Makes insulin. Insulin to regulate blood sugar. Right? So we have very specific reactions that are going on in these two organs. And so we're going to see some differences in hexakinase and glucokinase. So for right now, we're really going to focus on hexakinase. So hexakinase and glucokinase both catalyze a phosphorylation reaction. So this is one of those investments where we're using up an ATP. And what we're going to do is we are going to replace the hydroxide with a phosphate group. So that's on carbon number 6, so that we get glucose 6 phosphate. Now, when we do this, what's the charge on a phosphate group? -2. So when we are trying to add a phosphate group within an enzyme, that's a really negative group that we're trying to add. And so that can cause really big problems. So what do you think an enzyme is going to need in order to facilitate addition of a negative functional group? Some positive charges to balance it out. So hexakinase and glucokinase both require Mg2+ in order to be functional enzymes, because they need that additional positive charge inside of the enzymatic pocket to facilitate addition of the phosphate group and stabilization of your intermediate and all that kind of stuff. Okay. All right, so now we have glucose and it's been phosphorylated. So when we talk about blood sugar, right? What form is that sugar in? Is it glucose or is it glucose 6-phosphate? It's in the glucose form because it's out in the bloodstream, right? So how does this glucose enter the cell? How does glucose get into the cell? What's that? Not necessarily receptors, right? Receptors are like insulin. So if insulin binds to a cell, right? Then it's going to tell the cell, hey, you need to pick up glucose. So how does that work? You're going to have a transport, right? You're going to have to transport the glucose from outside to inside the cell. Okay. When we have glucose in the native form, it can go through these transport channels from outside to inside the cell. But what happens when we add a phosphate group to it? It now becomes negatively charged. Once it's negatively charged, guess what? It can't go back out. It's stuck inside the cell. So is this a really important mechanism for retaining the sugar inside the cell? Super important, right? So is it worth the ATP investment? Absolutely. So when we go to measure somebody's blood sugar, we want to measure what's actually out in the bloodstream, not what we're measuring inside the cell. So if we want to measure blood glucose levels, right? This is the thing where you can prick your finger and you can monitor your blood sugar levels. So you take a blood sample and that glucose that's present inside the bloodstream reacts with an enzyme that's on the test strip. And it's called glucose oxidase. And glucose oxidase, what's really important, will in the presence of glucose convert oxygen to hydrogen peroxide. Now that you have hydrogen peroxide, you have a substrate for peroxidase. So you convert a substrate here that's reduced into an oxidized product, and you can actually measure this level with this monitoring device. So this is how the newer glucose monitoring goes, right? Well, that's nice, right? This is how we can do it outside of the body. But guess what? Your body needs to monitor glucose levels for you. So you have a system inside of your body that's monitoring glucose. So we want to keep basically our blood glucose concentration at about 4 or 5 millimolar of glucose, right? So this is our normal blood glucose concentration. Look at our two enzymes, hexakinase and glucokinase. Hexakinase is basically everywhere except in the liver and the pancreas, where glucokinase is located. Tell me about hexakinase. When is hexakinase active and working? It doesn't matter how much glucose you have. You could have a tiny amount of glucose, right? The Km is 0.1 millimolar. 0.1 millimolar. That's tiny. So this is a very, very high binding affinity. So if glucose is there, hexakinase is phosphorylating it, right? And that's exactly what you want in the majority of your cells. But look at glucokinase. Is glucokinase the same? No. Glucokinase has a much higher Km, which means it has a much lower binding affinity. Why would you want it to have a lower binding affinity? Well, you only want glucokinase to be active if you've eaten a lot of sugar, right? If you barely take a bite of anything, do you want all the cells in your entire body to take glucose from the bloodstream? No, your blood sugar is going to tank and you are going to be in super big trouble. I only want to produce insulin when I've had a meal, when I've had a substantial amount of carbohydrate. Usually a lot of times the problem with that is people will take too much insulin or they will take not enough insulin. So it's that you didn't do the calculation right on how much carbs you actually ate.
No, but when you said that when it takes a little bit, if it was to take just the very little bit and take it, then your blood sugar would tank. Is that what happens?
No, usually people give themselves too much insulin. And so then the cells take in too much glucose from the bloodstream. Yeah. Okay, so we have glucokinase that's acting only basically when we have lots of glucose around. So what are the two things that we want to happen when we have a meal? We want to produce insulin and tell our cells, hey, you can take in the blood glucose levels. But the other thing is, what if I eat a carb-heavy meal? I'm going to have excess carbs. What do I do with that excess carbs? Store it in glycogen in the liver. So the pancreas makes insulin, the liver makes glycogen. What are the two organs where we have glucokinase? Liver and pancreas. So you need to be able to explain why there are differences between hexakinase and glucokinase, where they're located, and why that difference is important. Okay. All right. Now, we go on to our second reaction, phosphoglucoisomerase. What does an isomerase do? So you keep the same molecular formula, but you change the three-dimensional orientation of the atoms, right? So when we start out with glucose 6-phosphate, how many members are in the ring? We have a 6-membered ring, right? So now what happens when we go from glucose 6-phosphate to fructose 6-phosphate? Now we have a 5-membered ring. Well, you remember how the very first step was to phosphorylate carbon 6, right? Now, do you see how carbon 1 is going to look the same way that carbon 6 did before we phosphorylated it? So can you guess what the next step is going to be? The next step that we want to do is phosphorylate that carbon 1, right? Now we have basically a mirror image, right? You could cut it down the middle and it looks like a mirror image. Both carbon 1 and carbon 6 are now phosphorylated. So that happens with phosphofructokinase 1. You'll see it as PFK1. If I could speak and write the same things, that would be wonderful. Oh my gosh. PFK1, right? You'll see that in the book. They'll abbreviate it as PFK1. They don't necessarily tell you that, right? So PFK1 is going to do that same thing where you have a hydroxide and you're going to replace that hydroxide and you're going to put in a phosphate group. So this is another, guess what? ATP investment. So now we've invested a total of 2. So tell me, what was similar about our PFK1 reaction, our phosphofructokinase 1 reaction, and our hexakinase reaction? They both use up ATP in a phosphorylation reaction, but remember that table that I showed you just a few minutes ago? Is there a similarity between hexakinase reaction and PFK1 reaction? They are both irreversible. They have very negative delta G, so they are irreversible. Right? So let's go and add that to our PFK1 as well, right? So this is very negative delta G, so it is irreversible. Do you have to remember exactly the negative delta G value for these reactions? No. But do you have to know the 3 that are the highest? Yes, you do. Okay. All right, so now we have fructose 1,6-bisphosphate. So we have a phosphate group that's attached to our 2 different carbons. And this is the point where we're actually going to cleave this one molecule into 2 different molecules, right? So we're transitioning from the investment stage to the ATP production stage, right? Okay, so this reaction is catalyzed by aldolase. So when we look at this, what's actually happening, you remember how we said sugars can be in the ring form, the cyclic form, and they can be in the linear Fisher diagram form, right? Well, what actually happens is that this first linearizes and then it gets cleaved. So when we do that, you're actually going to have a linearization here So you're going to lose this bond here. Then you're going to have a cleavage reaction right there. So technically, to get from a cyclic to 2 straight chains, you would actually have to have 2 cuts. But aldolase only makes 1 cut, but it's to the linear form, if that makes sense. They don't, the book doesn't say that very clearly, which I wish they did, but they don't. So when we do this, there's a small problem. Our 2 products are not exactly the same. So we have glyceraldehyde 3-phosphate. So if we have, and then we have dihydroxyacetone phosphate. So look at the 2. One of them is an aldehyde, one of them is a ketone. Which one's which? Glyceraldehyde is the aldehyde, and dihydroxyacetone is the ketone. You can't use both of these substrates in glycolysis. You can actually only use one of them. So which one are we going to use? Look at your map. Which one do we need to use to get to the next stage? We want glyceraldehyde 3-phosphate. This is the one we want. So what we're going to have to do is do a second step with the dihydroxyacetone. So we have one product that's great, and we have one product that we need to convert. So we're going to convert our dihydroxyacetone into glyceraldehyde 3-phosphate using an endiol intermediate. So we're going to use an isomerase, right? We're not actually going to change the molecular formula. We're going to change the structure. So we want to get glyceraldehyde 3-phosphate. So what do we have to do? We just have to convert where our double bond was here between carbon 2, right, and the oxygen, to now being between carbon 1 and this oxygen. So all it is an isomerization so that we can generate glyceraldehyde 3-phosphate to continue. So that's why you see in the pathway in front of you, right, that one pathway makes glyceraldehyde 3-phosphate, then the other side makes dihydroxyacetone phosphate, then makes glyceraldehyde 3-phosphate. Yeah? Okay. So this is our end of our ATP investment, right? Once you have 2 products that are glyceraldehyde 3-phosphate, we are done with investment. All right. Reaction 6. Now we have glyceraldehyde 3-phosphate, and we're going to use this enzyme called glyceraldehyde 3-phosphate dehydrogenase. What are we going to do here? Be careful. It says dehydrogenase, but what's happening to the carbon-containing molecule? We're going to phosphorylate it. We are absolutely going to phosphorylate it. But when we phosphorylate it, what happens? What do we lose? We lose a hydrogen. Where does that hydrogen go? That hydrogen goes to NAD+, and we're going to make NADH. So you're going to make actually 2 NADH molecules per glyceraldehyde 3, per your pair of glyceraldehyde 3-phosphates, right? So you're actually doing it twice because you have 2 glyceraldehyde 3-phosphates. Yeah. So why is this important? This is really, really important because you are regenerating your NADH. And NADH is going to be super important in lots and lots and lots of different pathways. So it's important to keep track of where you're producing ATP and where you're producing energy electron carrying molecules, which is NADH. NADH is one of them, right? What time is it? All right, we can end there. We're on reaction 7. Y'all remind me, we ended on reaction 7. So we just got to finish it, and then we'll talk about regulation. This will be good. All right, anybody on Zoom have any questions? Y'all have a great rest of your day.
The homework is due tomorrow?
No, it is not. So when I added the extra day, the homework is not due tomorrow. When I added the extra day, I didn't go back and change some of the homeworks. I don't know how I missed them, but I went in and fixed them this morning. So they are not due tomorrow. Absolutely not. Absolutely not. I appreciate y'all telling me about it. Okay. So I'm guessing everybody on Zoom's okay. Let me end this there.